Thanks. So this is actually a small topic change from what was originally listed on the schedule. We made a slight change in company plans right after I submitted the abstract. So I think on the schedule, it's the talk title is referencing cancer diagnosis and lung cancer. But instead, I'll be talking about something, at least from a data perspective, that's similar, which is segmenting hearts in cardiac MRI using deep learning. And specifically, I'll be talking a little bit about how we deal with some clinical data that we're using, which is distributed in a sort of an inconvenient way, and how we work around that. So let me give you a little background first on how Arterys works as a company and our current platform. For the use case of cardiac MRI, typically the first step is the patient will receive an MRI in a clinic or hospital, cardiac MRI. The data is then sent to our cloud where it's stored and processed and reconstructed. And the cloud performs a few different functions. In addition to reconstructing and storing the data, we use GPUs in the cloud on Amazon AWS for rendering the data. So when the clinicians are using our web app, all of the rendering is done on GPUs in the cloud, and the user can use a very thin client, such as their phone, with very good performance. And also we use GPUs, obviously, for deep learning. So this is either as a pre-processing or online step of doing inference in order to detect features, for example, in the data. So to specifically color what we're using deep learning for, it helps to have a little bit of background in cardiac anatomy. What I'm showing here is a cross-section of the heart. In blue is deoxygenated blood. That is blood that is going to the lungs, ready to receive oxygen. And the red is oxygenated blood that has come from the lungs and is ready to be distributed through the rest of the body. So initially, deoxygenated blood comes from the body as a whole through the vena cava, superior and inferior, into the right ventricle. And as a caveat here, in standard cardiac views in MRI, the right heart is on the left on the screen. So they're reversed here. It's as if you're looking at the patient face on. So deoxygenated blood comes in here to the right atrium, flows into the right ventricle. The ventricles are the actual pumping parts of the heart. The atria are staging areas. Pumped from the right ventricle into the lungs, comes back into the left atrium, left ventricle, and the left ventricle is responsible for pumping it to the rest of the body through the aorta. So in a lot of different types of cardiac disease and dysfunction, the ventricles are the region of the heart that are affected, the pumping regions. So these are a particularly important area for performing segmentation on and for quantifying the volumes. The specific type of cardiac MRI that I'll be talking about is called SSFP, which is a specific kind of MRI pulse sequence that stands for steady state free precession, which I put up here because I knew I would not remember the acronym live. It looks like this, and just as a side note, if you look up Arterys online, you'll see a lot of references to 4D flow, which is a novel type of pulse sequence developed in collaboration with Arterys, Stanford, and GE Healthcare. I won't be talking about 4D flow here. That's a newer type of cardiac MRI, but because a lot of institutions still use this legacy method, SSFP, we are working on products to support that legacy data type. So I'll be talking about this kind of MRI. Right over here, I should say a typical SSFP acquisition consists of slices along the short axis of the ventricle. So if you remember from the previous slide, maybe I'll jump back for just a second. The heart has a long direction and a short direction. So these slices, come back one more time, are cuts like that through the ventricles. In the center here, the rounder region is the left ventricle. This is the part containing oxygenated blood. The crescent-shaped structure over here is the right ventricle, which has deoxygenated blood en route to the lungs. And then the darker region surrounding the left ventricle is the myocardium, which is the fairly thick muscle surrounding the left ventricle that is pumping the blood throughout the body. So one thing that you can see here that's going to be important later is that although the left and right ventricles mostly pump together, they're slightly out of sync, which means that the time point at which the left ventricle is completely contracted can be slightly off from when the right ventricle is completely contracted, and vice versa. The time point when they're maximally dilated may be different. And the reason this is important is because when measuring cardiac function, the radiologist will typically only segment and determine volumes at a maximum contraction and dilation. So the time points at which we have segmentations can be different for the left and right ventricles. And I'll explain why that's important next. So the end effect of these time points being a little bit different is that our data is not really in the optimal format. So there are three kinds of segmentations that we're interested in that radiologists typically perform in order to measure volumes within the heart. There's the endocardium of the left ventricle. Endocardium means basically the blood within the left ventricle. There's the epicardium, which is a contour that surrounds the entire ventricle, including the muscle. And there's the right ventricle endocardium, again, just the blood in the right ventricle. And typically, radiologists do not segment the myocardium of the right ventricle because it's very thin and difficult to see in these scans. So as a consequence of the fact that radiologists typically do not measure all of these contours in the same slice, and also the left ventricle epicardium is typically of lesser interest. It's only interesting in certain more esoteric cases. We do not have all three of these segmentations on all of our images. In fact, although we have left and right ventricle endocardium segmentations on most of our images, we have epicardium segmentations on far fewer, about 20%. And then when you take the images that have all labels, that's only about 18% of our total data volume has all labels. So this poses a problem if you want to use a very simplistic segmentation neural network architecture, because most images do not have all the labels you care about. And we want to avoid sort of the onerous process of making different models for each of the different labels, because all those models would take 3 times as long to train, and they would share a lot of information, or they would have a lot of information that could potentially be shared that wouldn't be shared. We wouldn't be exploiting the similarity in these problems. But let me show you an architecture that we implemented to do this the naive way and only use the data for which we had all 3 segmentations on any given image, which was only about 20% of our data volume. And then I'll show you some results of that and a way we improved upon that. So in this case, we have a standard convolutional neural network architecture. We have more than 3 layers. I'm just showing a cartoon over here where we have the data coming in from the left and then output predicted masks coming out on the right. And I just want to actually take a step back. How many people in the room have worked with convolutional neural networks in the past? So it's about a third or a half. Okay, so for the rest of you, I apologize. Some of this might be esoteric, but I'm going to show some pretty movies, so we can pay attention to those. All right, so the input into this model that's being trained is an image, which is a single cross-section, a single time point, a static image of the heart, and then a ground truth mask that is indexed, where every pixel is assigned one of 4 labels: background, and then the 3 classes, which take too long to say. And then what the network will do is it will produce probability maps for each of those 3 classes. So for every pixel, there will be a probability given to each of the 4 classes, and those probabilities sum to 1. And then when training, this data comes in, the model makes a prediction, it's compared against the ground truth, losses, backpropagated, et cetera, and then at inference time, the model actually can create these masks, which will be, again, probabilities, but can be, you can take the argmax function and you end up with these static masks like that. Again, the problem with this approach is very straightforward. It's a very standard classification architecture, except with segmentation, so by classifying each pixel. And the problem is that we only end up using 20% of our data, which fits into this format. So here's the result of segmentation when we use this architecture with 20% of our data. And what I'm showing here are probability masks colored according to the different classes, and then derived contours from them where we've thresholded the masks and then just drawn a contour around. And like the first thing that's going to strike you is the red contour, which is the right ventricle, is very poor. If we think the goal of this whole procedure is to deploy the results of this model in our app where a radiologist will click a button and these contours will be created automatically. And you can see the right ventricle in this case is not deployable. This would not be usable in a production app. And here I'm not talking about an FDA clearance issue because the radiologist has the opportunity to correct everything before determining clinical care. It's really a UX issue where the radiologist would essentially have to start from scratch with a right ventricle segmentation like this. The left ventricle segmentations look better, which is partially due to the fact that the left ventricle is a simpler shape, but still there's some noise there and some corrections would have to be made. So now I want to talk about the architecture, how we changed the architecture to allow us to train on all of our data. Now, again, well, the architecture has changed here so that instead of outputting 4 classes, we essentially interpret each class independently, but still within the same architecture, where we predict on each class versus the background of that class. So each pixel is either right ventricle endocardium or not. And those are all done separately for the 3 classes. So in this case, the training data is separate masks for each of the 3 classes, and loss is calculated separately for each of the 3 classes. And then the loss is summed across the 3 classes and back propagated. Again, the only difference here from a practical standpoint is the output. The internals of the network are the same. And the key advantage here is that if a given image is missing one of those 3 classes, we simply ignore that component of the loss. The network, when doing training, the network will still make predictions for a given training batch, even on a label for which we do not have a ground truth mask, but we will simply discard those predictions when calculating the total loss, sum the loss from the 2 classes that we do have ground truth on and backpropagate that. And in doing that, every image that has at least 1 ground truth contour can be used in the training process. So here are the results when we train it using all of our data, using that architecture I just described. You can see immediately the right ventricle contour looks much better. It much more closely follows the actual boundary of the ventricle, and the left ventricle contours look better as well. So the huge difference here is really from a user experience standpoint. Either way, you could deploy the model. You're not going to get in trouble with the FDA because the radiologist has the opportunity to fix anything that went wrong, but the radiologist needs to do much less work in this case to get to an acceptable segmentation and an accurate volume estimation. So that's a big advantage of this technique and why we really want to make it very accurate to ensure they have to do as little work as possible. And I just showed one example case there, but if you look at a bigger picture, the whole point of these segmentations is to measure ventricular volumes. So what I'm showing over here is specifically for the right ventricle, which is outlined in red in the previous slide, the relative volume error. So we have ground truth for, in this case, we're using a validation set with something like 100 cases, and we have ground truth there for what the correct volume is, and then we've predicted a volume. And what you can see here, over here on the left, is the model where we only use 20% of the data, the more classic classification architecture. And on the right in green is the model where we used all the data, the somewhat more sophisticated architecture. And you can see that we have a median volume error of about 0.3. That means about half of all cases have 30% or more error in their volume. Or if your glass is half full guy or girl, 30% have, man, this is too complicated. I think you get the idea. 30% error in half the cases. And when you go to the model that uses all the data, about 10% error in half of cases. So you've reduced the median error from 30% to 10%, which is really a huge savings. I mean, I think it's pretty obvious from this plot that this is a big advantage and is going to work very well. So that's the last content slide I had, and I just want to say the conclusion here, maybe this is not a surprise and it's not something you needed to be told, that more data is better, but the real conclusion from the effort that we went through to exploit that data is that the effort is often worth it. That data, our original version of the network was sort of locked away and it was challenging to access. So by putting in a little more effort, modifying the network architecture to accommodate it and to exploit all the data, we got a much better result, which is really nice because that means radiologists will have a much better experience using our product. That's the end of the talk, and I just want to mention that we are hiring. If you are interested in computer vision in the medical domain, we've got some really fun stuff for you. Go ahead and send me an email. We'll get in touch, talk more, and thank you all very much for your attention.
Thanks, Dan. I'm going to abuse my position as microphone holder and ask a question. I noticed that looks like you pretty much nailed it with the second model, but in the first model, the boundaries look quite smooth in the space dimensions, but they're kind of flickery in the time. Did you train on individual images or on the sort of 4D volumes?
Maybe I'll just go back to that. I think you were referring to this video or to either one?
Yeah, that guy.
So this model was trained using two-dimensional images only. So in this particular model, we have only used a single image at a time in training and inference. We're also experimenting simultaneously with using adjacent images in space and time, and also 3D convolutions, but this does not exploit that.
Cool, thanks.
Any other questions?
Can you talk a little more about the stack? Can you talk a little more about what you used, for CNNs?
Sure. A little bit more about the framework and model architecture. Sure. So we use TensorFlow for both our training and inference. So the whole goal of this is to integrate into the app. So TensorFlow is a good choice for us because of how much attention has been paid to using it in products. So we want to use this in a real product, a healthcare product. So that robustness was very important to us. And then in terms of the actual model architecture, we use a variant of the semantic segmentation architecture posed by Long et al. In 2014, which was, I think, the first paper that mentioned using a fully convolutional neural network for doing segmentation. So we have no pre or post processing of the images. The segmentation is wholly contained within the neural network.
I have another question. When you didn't have the labels for particular parts of the heart and you added that data, you didn't train those nodes, or how do you show that X? So that means you're not training the connections to those nodes, or you're just pruning them out?
So when training and this data comes into the network, this ends up being set to zeros, which is okay because it's ignored anyway. Okay. And we essentially have a flag, which is sort of just how we arrange the data, to indicate to the last layer of the network, which is right at the loss function, that we do not have information there. And then within the TensorFlow graph, we actually just delete the components of the loss. The loss ends up being a vector that has the same length as the total number of pixels that came in. And we just say that the components of that vector that correspond to that missing mask, delete them, take the average of the remainder, and backpropagate that. Does that answer your question?
Cool. Thanks for a great talk, Dan. And if there are any more questions, hopefully you'll be around afterwards, offline.
Yep.
Cool.
Thank you.
Thanks.